Hey folks, and welcome to another edition of Zog Science. Today we're going to be beginning our discussion about photosynthesis. We're going to start with the light reactions. So just a little bit of vocab review from some things you probably have heard before. Autotrophs, which you should have learned about it in Bio 1. Those are your self-feeders. They can feed themselves. A photoautotroph is a light self-feeder. There are also chemoautotrophs that can use chemicals to self-feed themselves. There are a lot of different organisms that have the ability to do, who are photoautotrophs, who can do photosynthesis. We've got plants, multicellular algae, you've got unicellular eukaryotes, cyanobacteria, as well as purple sulfur bacteria. So lots of different types of organisms that can do this. The purpose of photosynthesis is to convert light energy into chemical energy in the form of food, and this is going to occur within the chloroplast. Let's take a little bit look at a leaf structure. We're going to be using plants as our model organism for photosynthesis. Inside the leaf, there's kind of a sandwich thing going on. You've got the outer layers, and then in the middle, you've got the mesophyll, which is where the actual chloroplasts are in cells. And that's where photosynthesis is going to occur. You've got veins that are bringing water and nutrients to the cells, and then that's where the food that is produced is going to leave. And then you've also got the stomata, which is where carbon dioxide goes into the leaf, and then oxygen comes back out. Inside the leaves, you've got, again, those mesophyll cells, and they have the chloroplasts inside, and the chloroplasts have their own specific structures. Now, when you took Bio 1, you learned that the equation of photosynthesis was, 6CO2 plus 6H2O plus light energy yields carbon dioxide, or yields us oxygen and glucose. In reality, what actually happens is that we don't produce a 6-carbon sugar, we actually produce a 3-carbon sugar. And there are a lot of different ways that we can write this, but I just kind of wanted to highlight the idea that in reality, we don't produce that glucose molecule itself, we produce a smaller molecule which can then be packaged into glucose. So the question that was on a lot of people's minds is, where does the hydrogen come from? And where does the oxygen come from? And it was determined that the water was being split to produce electrons and the products of the reaction. So the hydrogen is leaving the water; it's going into the sugar molecule that's being produced. It's also going into some of that water that's being produced, and then you've got the oxygen as well. Photosynthesis is a redox reaction. We've talked about this before. The carbon is being reduced into the sugar, and then the water is being oxidized into the oxygen. There are two main stages of photosynthesis. We've got the light reactions, also known as photophosphorylation, and then we've got the Calvin cycle, which is carbon fixation. Photophosphorylation, we've talked about this before, right? Phosphorylation is the idea of us taking ADP and turning it into ATP. We've talked about substrate level. We've talked about oxidative. Now we're going to talk about photophosphorylation. So we need to talk about sunlight, because sunlight is what is providing the energy for this reaction to take place. The sun produces the full spectrum of electromagnetic radiation, all the way from gamma rays to radio waves. Visible light is what plants use to actually drive photosynthesis, which is only a very narrow band of the magnetic spectrum. When we talk about light, we need to talk about pigments, because pigments are what actually absorb the light. Pigments absorb some wavelengths while reflecting others. So the chlorophyll A and chlorophyll B are the two main pigments found in chloroplasts. We also do have carotenoids. Chlorophyll A and chlorophyll B absorb blue, orange, violet, and red colors, and they are going to reflect green colors, which is why plants turn out to be green. Then they also allow the green colors to go through, because they are, they don't absorb that particular sunlight. Carotenoids absorb violets and blues. So what happens is that we've got chlorophyll A, chlorophyll B, and carotenoids. They are all absorbing the spectrum over here. They are also absorbing a little bit of the reds and oranges and yellows. So what that means is that they're not really absorbing greens very well. And so when they're not absorbing it very well, that's actually allowing that light to get through. This is what chlorophyll A and chlorophyll B look like. They have a slightly different functional group right here, but the vast majority of the molecules look the same. This is that molecule that was on the test, on the very first test we took, that was kind of scary looking. You notice that there's a magnesium right here. The magnesium is very important and allows the chlorophyll to perform its function. If plants don't have magnesium, they would not be able to do photosynthesis. So when a photon of light strikes the chlorophyll molecule, it causes one of the electrons to move to a higher energy shell. Okay, this is going back to our chemistry, and that is now in what is known as an excited state. As it is falling back down to its ground state, so it gets excited, it falls, it goes up. As it goes back down, heat and energy are released, and that heat and energy is then used to drive the process of photosynthesis. And magnesium again helps to facilitate this in the chlorophyll molecules. What this means is that if we were to isolate chlorophyll and just have it by itself, we could shine light on it and it would fluoresce. And this is the color that it would be. It's kind of this weird orangey red color. So what is actually absorbing the light in the photosynthesis? What we have are these photosystem structures. These are located in the membrane of the thylakoid, which are those little stacks of things inside the chloroplast that actually contain the chlorophyll. They are a light harvesting complex. They're made up of pigments and proteins. So you've got chlorophyll A, chlorophyll B, as well as your carotenoids in there that are actually absorbing the light. And then you have the reaction center complex, which is a special pair of chlorophyll A, which are the primary electron acceptors. So what's going to happen when a photon strikes one of the chlorophylls is that's going to excite an electron, which is then going to fall back down, and it's going to transfer its energy. And that energy is eventually going to get sent over to the chlorophyll A molecules. The chlorophyll A molecules will accept that energy, and it is then going to boost up a specific electron, and that electron is going to meet the reaction center complex. That is then going to, it's going to boost the electron to the electron acceptor, which is going to take that electron and is going to hold onto it. All right. So basically, what we're doing is we've got our photon. It's hitting a chlorophyll. It's bouncing it around. The energy is being transferred, not the photon itself, but the energy from the photon is transferred from one chlorophyll to the next. Reaches our special pair of chlorophyll A molecules, which boost an electron up to the primary electron acceptor, and that's the whole job of the photosystem, which is what we just talked about. So, two different photosystems are going to work together. We've got photosystem 1, which has P700 chlorophyll A, and we've got photosystem 2, which has P680 chlorophyll A. These are pretty much exactly identical to chlorophyll. However, they have slightly different proteins in the photosystems themselves that shift the absorption spectrum. So what that means is that chlorophyll A absorbs things best at the 700 wavelengths, and chlorophyll A absorbs things best at the 680 wavelengths. So they are going to work together, okay? This process requires both of them. What happens is that light is captured by the photosystem 2. The electron is boosted up to its primary electron acceptor. Electrons from water is split apart into oxygen gas, hydrogen, and electrons. Those electrons are then used to replace the electron in the P680 that boosted up its electron to the primary acceptor, right? Because it's lost one to the primary acceptor, it needs to gain more back. So those electrons are used to replace that. The oxygen and the hydrogen are released into the thylakoid space. So here's where oxygen is being produced in our photosystem. The electron that is now in the primary acceptor, it is going to be sent down an electron transport chain. And again, it's going to be moving from areas of low electronegativity to areas of high electronegativity, and that is going to produce ATP. And it's going to produce it using chemiosmosis and a proton gradient, just like we learned about when we talked about the mitochondria. That electron that has now gone down the electron transport chain is going to reach P700 in photosystem 1. All right, so this is where you kind of have to think about this and remember this, is that it starts in photosystem 2 and goes to photosystem 1. The reason being they discovered photosystem 1 first, and then they discovered photosystem 2, so it just got named backwards. But more light energy is used, captured using the chlorophyll. It bounces around, and then it goes to the P700. It excites that electron and bumps it up to its primary electron acceptor, which goes down another short electron transport chain that then drops it off at a NADP plus reductase, which is an enzyme that helps to form NADPH. NADPH is very similar to NADH. It does the same function of holding electrons. It's just what's used in plants as opposed to NADH. And it takes NADP plus and it combines it with hydrogen in order to form that NADH. Again, The whole point here is that we're taking the electrons and we're using them to form this NADPH, just as the same thing was going on when we were talking about the mitochondria and its function, right, of like the Krebs cycle, things like that, were to produce the NADH. Now we're producing NADPH. So just to kind of look at this in a little bit of analogy, we've got a photon that is boosting up an electron using photosystem 2. That electron then goes down this mill, it produces ATP. The electron is then boosted up again by another photon, and that is then used to produce NADPH. The light reactions, again, remember that taking place in the thylakoid space. Inside that thylakoid space, we are building up a concentration of hydrogen. That concentration of hydrogen is used to help drive ATP synthase to produce ATP. And then NADPH and ATP are both going to be sent along to the Calvin cycle, which is also sometimes known as the dark reactions, because it doesn't require light to occur. So this is the only part of photosynthesis that actually requires light in order to function properly. It again uses a very similar system to what is going on in the electron transport chain, where we are trying to use a chemical gradient to produce ATP, and then also we are using electrons moving from areas of low negativity to high negativity to eventually produce NADPH. So there are a few comparisons we can obviously make between the mitochondria and the chloroplast, particularly when it comes to this part of it. We've got some sort of electron transport chain that is driving ATP synthase using chemiosmosis, producing ATP. A couple of things that are different. The electrons in mitochondria come from food, while the electrons in chloroplasts come from the water. And that water is then split apart. This is why water is so important for plants. And then mitochondria use energy from the food, whereas chloroplasts use the photons from light that they are bringing in. So that's kind of your beginning introduction to photosynthesis. We'll talk about the Calvin cycle and the dark reactions in our next video. So thanks for watching, and I'll see you next time.